Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Welcome back to UI Creates 2024. First of all, let's say thanks to Allah Subhanahu wa Ta'ala who has been giving us happiness, health, and mercy so we can attend in this event today. Salawat and salam always be delivered to our Prophet Muhammad Sallallahu Alaihi Wasallam who has guided us from the darkness until the brightness like now. The honorable, all participants, and all of the lecturers. Today, we will continue on our lecture about drug target and drug target interaction with Ibu Samira Husain Alamudi, PhD. Will be moderated by Bapak Dewangga Oki Bagus, SSI, MSI, PhD. Bapak Dewangga Oki Bagus, SSI, MSI, PhD, is a lecturer at Chemistry Department, Universitas Indonesia. He has research interest at green synthesis of nanoparticle, phytochemical screening of plant extract, and electrochemical water splitting. He got bachelor degree from Universitas Negeri Semarang, Indonesia, master degree from Chemistry Department, Universitas Indonesia, and PhD degree from Universitas of Kyushu, Japan. She had many science publications and awards. One of them is Max Scholarship by Japan. Ladies and gentlemen, I will give time to our moderator, Bapak Dewangga Oki Bagus, SSI, MSI, PhD. To Bapak Dewangga Oki Bagus, SSI, MSI, PhD, time is yours.
Yeah, thank you very much, Miss Hilna, for your nice introductions. Greeting from Indonesia. Good morning, special honorable guest today, Dr. Heri Cahyana, Dr. Nurdini, Dr. Bayu, and also Dr. Samira Alamudi today. Today for about 2 hours later, I will be with you as a moderator with an interesting topic about drug targets and drug target interactions, who will be delivered by Dr. Samira Hussein Alamudi. And previously, as we can see that research on drug delivery and also including drug target interaction has considerably attracted many researchers in the past decades, specifically in the field of medicinal chemistry for our life, and strongly remember in our memory of 4 past years ago how tragic catastrophe of COVID-19 pandemic destroyed all sector of our life, which requires the discovery of medicines. So even after this pandemic has been stopped globally across the world, now the researchers is still competing to develop the drug in the field of medicinal chemistry to be more effective for treating COVID-19 viruses. Okay, without waiting for a longer, I will let you inform about the wonderful curriculum vitae of Dr. Samira. First, as you can see about the email and mobile phone address for the student who has interest with the research background from Dr. Samira, you can further contact her. And Dr. Samira has research background in chemical biology, cell imaging, medicinal chemistry, molecular biotechnology. And Dr. Samira has graduated undergraduate in chemistry from Universitas Indonesia in 2009, and she completed PhD in chemistry and chemical biology from National University of Singapore in 2015. And she has so many experience. Now she has been appointed as assistant professor in the Department of Chemistry, Universitas Indonesia. And about 2022, she is a laboratory director, Genomic Solidaritas Indonesia, and also has so many experience in the research fellow and also research coordinator in the Laboratory of Agency for Science Research and Technology, Singapore. And Dr. Samira also has so many selected publications in so many reputational international journal. As you can see, there are so many scientific publications of her. Also, Dr. Samira has international patent about background-free fluorescence probe for live cell imaging, and Dr. Samira also has been awarded by several recognitions, specifically in the field of COVID-19. COVID-19 from A-STAR, Ministry of Health Singapore. This is one of award from Dr. Samira. So as you can see, there are so many awards she have been granted. Okay, without waiting for longer, okay, before we will listen the explanation from the Dr. Samira, I would like to remind the students, saya nak mau tengok those who have not open your camera for your student, please open your camera because our team will assess your complete attendance from your attendant with opening camera. So please, if you did not open your camera, please open your camera now. Okay. And also, one again, if there is any questions, actually we have Q&A session in the last part of our lecture online today, about 10 or 15 minutes in the last, but in the middle of explanation from Dr. Samira, you can have question or whatever you think, whatever you have a question, please ask in the middle during explanation from Dr. Samira. Well, Dr. Samira, how are you today? Is it okay to start?
Hi, Dr. Dewanga. How are you doing?
I'm good. Okay. Please take your time, Dr. Samira.
Thank you, Dr. Dewanga. Thank you for the very nice introduction, yeah? Hi, very good morning, everyone. It's nice to see you again. I see some familiar names. Now I see some on your camera, so it's good to see your face. Because last week I only see your name, like I remember Adam Hezron, right? It was a good discussion. Now I can see your face. It's nice. Yeah. Okay, so today we're going to continue our lecture from 2 days ago, yeah, not so long ago. So not yesterday, 2 days ago, so we learned about the physical chemical properties of the drug molecules, specifically the small drug molecules. Now we want to talk about what is the target, the drug target, and how the interaction between the drug and the target, right? So let me just share my screen. Okay. All right. Can you see my screen?
Yes, doctor.
Yes, doctor.
Okay, nice. Okay, so now we're going to discuss about this drug target and drug target interactions. So as Dr. Dewanga mentioned before, if you have any question in the middle of the discussion, so just raise your hand or you can just unmute yourself. Okay, so it's good to have a discussion. Right, so it's the same thing like 2 days ago. So I recommend you to read this book. Is available online. So I think everyone can access this book. It's the Medicinal Chemistry by Graham Patrick. So if you are interested in this field, you can have a better overview about this field. So it's a good reading for you in your leisure time, rather than you just scroll your phone. So yeah, every now and then it's good to read a book. So for today's, we're going to have some of the topics that we're going to cover. Of course, first is the introduction about the drug target. And then from the drug target, we move to how we can choose a proper or a correct disease to develop the drug and how we can choose a drug target. And then we're also going to discuss what types of the target inside the body, and then what are the molecular interactions happening when the drug interact with the target, and also, as like 2 days ago, some case study. Okay, first, as an introduction. So I think in the first lecture, yeah, if I'm not wrong, Prof. Zahir already give you the overview, the history about the drugs. So the ancient times, we don't really know the word or the term drugs. So the medicine, when you want to cure some disease, the medicine usually come from the natural products. Until now, even now we have this TCM, traditional Chinese medicine. Usually they are from natural products like herbs and all that. We didn't really know the active structures or the target of that medicine. We only know that when we take this medicine, we feel better, we feel good, right? But we don't really know what are the mechanisms, what is the target, and how the action inside the body, we don't really know. But later on, the serious effort were made, try to isolate and also purify the active compound from that natural products, from that herbs. So the natural products later became the lead compound, right? The lead compound, when you if you remember from the previous lecture, the lead compounds later on can go for further development, preclinical trials, clinical trials, and then can become a drug. So in this scenario, traditionally, there were no real design or like the reason why you choose this, right? So just you take these herbs, you feel better, then let's isolate and then see how the structure and everything. So no such design. And most of the discovery were made like a serendipitous discovery, like made by accident kind of thing. So the serendipity is actually when you can define it as a valuable discoveries accidentally found by the scientists. So it's like in the drug discovery point of view, it implies the finding of one thing while you are looking for something else. You know, you look for A, then you got B. This kind of serendipitous discovery. One of the famous example is the discovery of penicillin. So penicillin is the antibiotic, right? It was discovered by Alexander Fleming. So that time, Fleming was not looking for antibiotics. So it was kind of discovered accidentally when he was doing some culture of the bacteria, and then he found some fungus growth on that petri dish, and then he later discovered that this active ingredient is called the penicillin. Another famous serendipitous discovery was streptomycin. It's also the antibiotic for TB. It also invented from a random curative trials and trials of treating the TB, the tuberculosis, right? And it's isolated from the bacteria called Streptomyces griseus, hence the name streptomycin. So just a little bit story about this penicillin. So as mentioned, it was discovered by Fleming. Actually, he was studying this bacterium. So he tried to culture this bacterium on the petri dish, right? On the petri dish, but then he noticed, oh, contamination happened. Contamination happened. Normally, as a scientist or as a microbiologist, you see some contamination, you will throw that plate, right? Because that's not what you are looking for. You are looking for your very healthy bacteria that you are trying to grow. But then you found this ugly looking fungus, the bluish fungus, if you can see. Let me check. Let me see my pointer. Yeah. You see this ugly looking fungus that, you just throw your plates, right? But then not for Fleming. His curiosity brought him to the discovery of penicillin, because he later concluded that the fungus here, the blue ugly looking fungus, actually killing the powerful bacteria that he's trying to grow. So later he isolate the active ingredient from this blue fungus, and then he discovered that this fungus actually secrete this active compound called penicillin. So later on, a few years later, another two scientists tried to characterize this penicillin, and then in 1945, three of them has jointly awarded the Nobel Prize in Physiology for the discovery of this penicillin. So that's how things usually done long ago, right? So it's a little introduction again about the drug. So we know already that the drug is actually a compound, or you can say any entity that interact with a biological system in the organism, right? And produce a biological response, right? So it binds to something in your body and then it produce biological response. And so is poison, right? Poison also something that can interact with the biological system and it produces a response. The response can be positive, can be negative, right? In the case of penicillin, it's interesting. It is a drug, right? It's antibiotic drugs. When you got infection, you take the penicillin and it cures you, it heals you, right? But actually what penicillin did, it kills the organism. It kills the bacteria. Now, what do you think? Penicillin, do you think is a drug or a poison? Anybody wants to offer their opinion? Anybody? Drug, poison, right? Both are, say, chemical entities, any compounds, right? Both can be administered to your body. Both can interact with the system in your body. And both of them can produce biological response, whether it's positive or negative. Correct? So there's the similarities between drug and poison. But what is the fine line between the drug and the poison? And I give you one example of this penicillin. Penicillin, people consider it as a drug, right? But actually, the mechanism, it kills the organism. When you want to have drug should heal you, not kill you. So what do you think? Somebody wants to offer their opinion? Any opinion? It's a discussion, right?
Doctor, can you hear me, Doctor?
Yes.
Oh, okay.
Who's
So,
speaking?
Hezron. Hezron's here.
Your voice is too big. It's too.
Oh,
You speak a bit louder.
hello, hello.
Oh yeah, hi Hezron. Go ahead.
Okay.
What do you think?
So, I think penicillin has
Yep,
these
I can hear you.
both these things, which is drug and poison, because for us humans, it acts as a drug, which is like penicillin is an antibiotic drug because it kills the kind of organism like the bacteria in our body. But for the organism itself, like the bacteria or the virus, it acts as a poison for them.
Hmm.
So
Okay,
that's my opinion.
good. It's like a two sides of a coin, huh?
Yes.
Yep. Good, Hezron. Thanks. Anybody else wants to give their opinion? Not really give, to share. Share your opinion. What do you think? Do you
Just a
agree?
bit of just a bit of a question to more validate this. Does penicillin have any effect on our body, or is it just the bacteria?
It has effect in your body. So that's why when you have
Okay.
infection, the doctor prescribe you antibiotics, then you feel better, right? So it means
Okay.
that something is going on in your body.
Okay, so it's both acting on the organism and the body.
Correct.
Okay, so I would also have to agree with Hezron then, that it's both a drug and a poison, in
Yes.
the sense that it's a drug for humans, also poison for the bacteria.
Okay, good. Thanks, Adam. Anybody else? Okay, how to see the other participant, huh? Okay, I see some of the participants on the camera. Good to see you. Anybody wants to offer opinion? Okay, then I'll make it into assignment then. All right, so let's discuss this because we have a brief discussion, but you can make your argumentative essay because in this lecture, I also need to give you assignment, right, as a part of getting a credit for it. So you can make a short argumentative essay on this topic. Penicillin, is it a drug or a poison? What is the fine line between drug and poison? And when you have your arguments, and I want you to explain why comprehensively. So it's just short essay, don't need to be too long, maximum 1 page. Then you can submit to Dr. Bayu later. Okay. Anybody want any questions about this assignment? This little assignment, I would say. All good? Okay. Then let's continue. Oh, Dr. Dewanga, please remind me about this assignment, yeah? Because they need to submit to the organizer, Dr. Bayu.
Of course, Dr. Samila, yes.
Thanks. Okay, now we move to the drug design. So the ancient times, as we discussed before, the medicine were natural products. So there were no prior design. They don't know the target. They don't know the mechanism of action. But now, with the progress, with the development of the technologies and science, especially at the molecular technologies, there comes the word the drug design. It means you can design the drug from the scratch. When you need to design the drug, so you need to know what you want the drug to behave, right? So that's why it is called the drug designing. So the drug designing also called the rational design, because you need to rationalize the way you think about how the drug will behave in a certain condition. So the discovery process of finding the new medication is based on the knowledge of the biological target, right? It should produce the biological response in your body, but which part of your body? Your body is like very complicated system. So you need to know what is the biological target of your drugs. It involves the design of molecules that complementary in shape and charge, usually to the biomolecular target, in which they will interact and therefore they will bind to it, right? And now the advancement of the digital technology, the drug design now relies on computer modeling techniques as well. So this is just a refresh about the classical process in the drug development. So you know already, it started with the defining the target, right? The target validation, and then it moves all the way to preclinical tests, and then followed by the clinical trial, phase 1, 2, 3, up to the approval by the FDA. So, but the first step is you need to validate the target. Before you can validate the target, you need to identify the target first. Validation and identification is two different things, right? Before you can identify the target, so you need to identify the disease first. You need to know what kind of disease you want to cure, quote unquote. Right? So in approaching the approach in identifying the drug target, usually it goes to two directions: conventional approach and the modern molecular approach. The conventional approach usually it starts with the studies about the physiology, the clinical, the disease in animals and the human. And then it exploits the existing drugs with the known effects in some of the disease. So you can investigate the mechanism, but not really at the molecular level. But the modern molecular approach, it allows you to understand more about how the genes are altered and how that affects the protein. Usually the target is a protein, how it affects the protein that they encode in the body. So driven by the advances in the molecular biology and the Human Genome Project, it involves the new emerging field that you already may be familiar with, is genomics, proteomics, metabolomics, transcriptomics, so all the omics. So we call it the omics era. Right, and then as I mentioned, the first thing first you need to do is you need to select the disease. You need to choose what is the appropriate disease for you to design the target for the drug. If you are in academia, usually in academia, the professor, the scientists, they usually have personal interests, right? Because they have the academic freedom to do what they feel like it based on their curiosity. So it based on the personal interest. Usually they will choose whether the common or the rare disease. And it also depends on the available grant funding because, well, doing research is not cheap. So you need some of funding from the grant bodies. But from the point of view of pharmaceutical industry, it's slightly different, right? Pharmaceutical industry is already part of the corporate business strategy. So you cannot really follow the personal interests of the scientists. You want to follow the market. So the pharmaceutical industry, they tend to avoid products with a small market, right? They will check how is the disease prevalence, how is the likely revenue of the drugs, and what are the resources that they have, and also how is the number and the scope of the projects that they handle at the same time, and also what are their competitors. So they also need to check the competition on that field. So it's slightly different point of view in selecting the disease, the consideration for those in academia and for those in pharmaceutical industry. Okay, so the priority for the pharmaceutical industry, usually the question that they will ask whether they can profit from the marketing of the new drug, whether it's outweigh the cost of the developing and testing the products. It makes sense, right? Because you know already to develop a drug, it takes like 10 to 15 years and it costs, for the new drug, it costs 2 billion of USD, right? It's too much money. So once you want to develop a drug, you need to weighing whether the cost and the profit, whether you can get more profit of it, right? And then the questions to be addressed usually whether the disease widespread, like the cardiovascular disease, malaria, and others, and does the disease affect the first world, or whether the drugs already on the market, the competition, the competitors, right? And if so, what are the advantages and disadvantages if there are already competitors on the market, right? And whether one can identify a new market advantages for the new therapy or for the new drugs. And now we come to a more in-depth of these topics. How do the drugs act actually, yeah? How is the influence of the drug molecules on our cells? There is a saying from Paul Ehrlich. I cannot say in Latin, I hope it's correct. Corpora non agent nisi fixati. I'm not sure whether I say it correctly. They say a drug will not work unless it is bound. Bound to where? To something. Because for the drug to produce biological response, it should interact to something. That something, we call it target, right? All right, now choosing the drug targets. The drug should have a specific effects. Let's take a look at the penicillin example. The penicillin, it cures human, I would say, human, but it kills the bacteria. So it has some selectivity. It has some specificity, right? So the drug should have some specific effects. And this is more of a result where they act in the body, right? So they need to have also selective targets. So for the specificity and selectivity, we can characterize it into several characteristics. First is the target specificity and selectivity between species. For example, penicillin. Penicillin, it kills the bacteria, but it doesn't kill us, human, right? Selective between species. Second one is the specificity and selectivity within the body. So like specific enzyme inhibitors, it works on certain area in your body and it doesn't work on the other area of the body. And also the drugs targeting to specific organ, even though it has same family of the target, like receptor. This one is the example is beta 1 and beta 2 receptors, but it acts differently in different part of the organs or the tissue. For example, in the heart and in lungs, even though it's the same receptor, they will have a certain degree of differences, right? We want the drugs to be selective. Like, for example, this is a drug, some drugs, and we hope this to bind only to the target. We don't want this to bind to the non-target one, because what happens if the drugs bind to the off-target? What do you think will happen? Anyone?
Can I offer an opinion?
Yes, sure. Raihan.
If it goes off target, then it might actually affect the organs or the target that we don't want it to be affected, or it might actually cause the opposite effect instead.
Correct. Correct. In other words, if it binds to the off-target, it may have probably severe side effects. We call it side effects, right? Good. Raihan. Oh, I didn't see your face. Never mind. All right. So that's about how to choose the drug target based on the selectivity and specificity. Now we are talking about body. But inside of our body, we have trillions and trillions of cells, right? So now we have a look at, we look closer at the cell system. Cell is we call it, when we learn in elementary school or middle school, is the smallest unit of living things, kind of thing, right? But even though this is the smallest unit, it's the most it's a complicated system. It's a very complicated system. This is the cartoon representative of one mammalian cell. So inside your single cell is very small, like 10 micron in size. It's super small, but inside is very, very complicated. So you see several organelles here. Like you can see the nucleus in the middle. Inside the nucleus, you have nucleolus. And outside the nucleus, you have this Golgi complex. You have ribosome, lysosome, microtubules, and everything. And they are all very active and organized in such a very organized way, right? So if you see here, among of these organs, you also will see biomolecules, actually. Biomolecules inside here. You can see some organic molecules, you see some inorganic molecules. So it's a very dynamic environment. And one process may affect the other process. So that's about the cell. And if you look at even deeper, the surface of the cell surface, it is bound with a membrane. It's called the plasma membrane, right? We learned this a few days ago. So the main component of the biological membrane in eukaryotic cells, like mammalian cells, like human cells, it is called the phosphoglycerate structure. It's actually a phospholipid, but it has some glycerol functional groups. So if you look at this, it's the lipid bilayer, right? Phospholipid bilayer. And here, usually we have this protein embedded on it. So this phospholipid, usually it has the hydrophobic tails, and it has some hydrophilic groups, the polar head groups, we call it. And for the drugs, usually the drugs will bind to the target. That's what we want the drugs to behave. It binds specifically to one target, because this target associates with some disease. So we hope that the drugs should treat the disease because of this interaction. So the interaction of a drug with the molecular target, it involves a process called a binding. This binding, the drug binds to the target. And there is usually a specific area in the target, so it's not blindly just bind to any part of the target. It's not. It only binds to a specific area where this binding takes place, and it's called the binding site. We call it binding site. And at the molecular level, the target usually is specific biomolecules to which the drugs will bind. So the biomolecules that usually drugs be able to bind, there are four main molecules. There are proteins, nucleic acids, carbohydrates, and lipids, right? There are these four major biomolecules in the body. So if you look at statistically, the current drug targets, the majority of the drug, they target this enzyme, right? Enzyme is which part of this, it's already here. Enzyme is part of proteins, right? So almost half of the drugs, they are targeting the protein types called enzyme. And the second one, it binds mostly prefer the GPCR, is another type of proteins. And some numbers, it binds to the DNA and other type of biomolecules. So and also some of the statistics here I got it from the Nature Review Drug Discovery. Apparently, not all drugs binds to biomolecules, not everything. So you can see here some percentage it binds to, we call it miscellaneous drugs. What does it mean? For example, here the antacid, you know antacid, right? If you have some, what do you call, the problem with your stomach, right? The antacid, actually, it doesn't really binds to any biomolecules in your stomach. It targets some chemical, is like the modification of the chemicals that can adjust the pH in your stomach. And also another example is laxative. Laxative, it doesn't really interact with the gut in your body, but it actually interacts with the gut environment or the content of the gut. Right, let's see what are the molecular targets, the top 10 molecular targets in this previous year, yeah? So let's see. If you can look at closely the target type, I hope you can see my pointer. The target type, the top 10s are enzyme, transporter, receptor, GPCR, and others. These all are proteins. So they are targeting proteins. And how about the drugs here? If you see the drug, usually, not usually, all of them I see. All of them are small organic molecules. This is the common type of drugs molecules. Yeah, this number is ranked by cells, yeah, and based on the database in the US. But now, how about now? There is a shift in the molecular target. So if you can compare the previous one and the current one in 2014, there is the shift in the drug target and also the drug types. It is due to the advantages of the development of molecular technology. If we can just take a look closely, previously we see the drugs, they all are a small molecule, but now we have such as this called biologics. Biologic is not the small organic molecules, such as antibody. So if you see here, number 1, adalimumab, and then number 3, infliximab, number 6, rituximab, trastuzumab, and bevacizumab. All here are antibodies. So when you see this name, what one thing you notice about the naming? Anybody wants to give opinion? All the antibody. What things you notice about the naming of this antibody drug?
They all end with MAB.
Again?
They all end with MAB.
Correct.
MAB is always at the end of each one.
The MAP. Usually when the drug is developed based on the antibodies, they will end with the MAP. MAP is monoclonal antibody. The AB is the antibody, hence the name. So it shifted, like you see is 1, 2, 3, 5. 5 out of 10 of the drugs now are no longer not only small molecule, but it moves to the antibodies, for example. Another thing is like the recombinant protein, like number 2 and number 4. So the shifting is actually it gives the medicinal chemists a more variety of developing probably better drugs. So now let's see. Protein is a target because as you see before, the majority of the drug targets still the proteins. They actually used in a majority of the drugs, right? Such as the enzyme, receptors, transport protein. And this is just some of the example of the proteins that are embedded in the cell membrane. There are GPCR, there are ion channel and transporter. So what is protein? I'm sure all of you already know, but let's just refresh ourselves about protein. Protein, you can say, is actually a functional biomolecule that made up of amino acids, right? So you have this polypeptide. It's a sequence of amino acid. You call it the primary structure of proteins. And then it can have some hydrogen bonding of the peptide backbone, and it can form a very unique structure. We call it beta sheets or alpha helix. This we call the secondary structure of protein. And then this secondary structure can have three-dimensional folding due to the interaction from the side chain. This we call it tertiary protein structure, right? From here, this one structure, the tertiary structure, they can aggregate together and make a one functional protein called, and the structure we call it the quaternary structure. For example, this is the real cartoon representative image of a protein called human cyclin-dependent kinase. It's like this. So this protein, if you see this statistics, majority type of proteins that are being the drug targets are enzyme, right? 47%. So what is enzyme? Enzyme is actually, we call it the biological catalyst, right? To increase or the speed of the reaction inside the body. So the many important drugs act as an enzyme inhibitor. They hinder or prevent the enzymes acting as their natural behavior as a catalyst for the particular reaction. So during the normal condition, right? This is the enzyme, and you have the substrate. The substrate will bind to the active site or the binding site of enzyme. So the enzyme can do their biological process nicely, properly. But then when you add this inhibitor that can act as a drug, it can compete with the same binding site of the enzyme. So that's why the reaction here that should happen between substrate and enzyme will not happen. This is called the competitive inhibition. Another thing, it can be a non-competitive inhibition. So it doesn't compete with the active site of the enzyme, but it can bind to the other part, different part of the enzyme, but still, because this interaction, even though it's not the active site, even though it's not the binding site, but it will affect the structure of the enzyme. So what happened? As a result, this substrate cannot bind to this enzyme. So the enzyme cannot do this biological process normally. Let's see some example of how the drug was developed to target a specific enzyme. One example I put here is the AZT. AZT, I think I mentioned before in the previous lecture, yeah? AZT is one of the drugs for the anti-HIV. It's the HIV drugs. It's actually the antiviral agent, and it's made based on the analog of the thymidine. So you see here thymidine. Thymidine is one of the building blocks of nucleic acid. Yeah, building blocks of nucleic acid. We have 4: A, C, G, and T, and this is the T. So they only change this hydroxyl on the thymidine here in the C3 carbon atoms from hydroxyl change to azide. And it targets the HIV reverse transcriptase because it targets this transcriptase, so it prevents the elongation of the nucleic acid. So that's why, sorry. So that's why it inhibits the function of reverse transcriptase that the enzyme that the virus used to make a new DNA copy. So that's why the growth of the virus, the HIV virus, will be inhibited. That's enzyme. And then we have different type of protein called the transport protein. The transport protein is actually the integral transmembrane of the proteins. They exist usually within the membrane, the cell membrane. So it's embedded inside the cell membrane, and it can transport any substances selectively. So the drugs can be designed to take advantages of this transport system. It can be goes in two ways. First, it may give you the access to enter the cells. So the drugs need to enter the cells, right? Some of the drugs, they cannot pass this cell membrane passively. So they can take advantages of this transport system to gain the access from the extracellular goes into the intracellular. Or the other methods, the drugs can be designed to block this transport system. So this transport system cannot function properly. There are two types of transport proteins. In general, we call it the channel protein. It's the channel protein. So it has like this long channel for this any compounds to pass through this membrane. Because the compound cannot pass through the cell membrane passively, so it needs to go to this channel. So that's why it's called the channel proteins. And it can be a carrier protein, similar to the channel protein, but it needs to be bound to this carrier, and then it will transport all the way to the intracellular. That's the transport protein. So what are the examples of the drugs developed to target this protein? One example is the famous Prozac. Yeah, it's one of the very famous antidepressant drug to tackle the depression. The Prozac disease, the structure is a very small, simple protein, simple compound, right? So it designed as an inhibitor for this protein called the selective serotonin reuptake protein. So the protein is called the 5-HT1A. It acts as an inhibitor for this presynaptic serotonin. How does it work? The mechanism of action is by blocking the presynaptic reuptake of the neurotransmitter serotonin. Serotonin is the neurotransmitter, right? To give you certain feelings. The lack of serotonin in the brain, it has been linked with the depression. So when you want to cure, not cure, to tackle or to reduce the depression, you can do it by blocking its uptake, the serotonin, the uptake. So the serotonin that is released from your brain has a longer duration of action. So this is how the project was developed. The next one is receptor, different type of protein. So the receptor is a type of protein that can receive and transduce the signal that may be integrated to the whole biological systems. These signals usually is called chemical messengers, which can bind to this receptor and produce physiological responses, such as the change in electrical activity of cells. So the drugs can behave in two ways. They can behave agonistically, or we call it agonist, to activate the receptor, or the other way around, antagonist, by blocking the receptor. So it's two ways, yeah? When the drugs behave as an antagonist, it blocks the receptor and it prevents the natural messengers from the binding. When the natural messengers cannot bind to this receptor, so the signal cannot be passed through to your biological system, the whole biological system. One of the very famous receptor that can be targeted for the drug development is called GPCR. So like if you key in GPCR, there are so many drugs, so many papers, so many research has been conducted to designing the drugs to target this GPCR. GPCR is actually a G protein coupled receptor. It's a type of protein. It's a type of receptor. It's one of the largest family of human membranes. If you see the structure here, it has 7 membrane subunits, and it is linked to the intracellular part, which can interact when activated with the G protein. So you see this very unique structure. If you see, it's like 1, 2, 3, 4, 5, 6, 7. It's the 7 subunit, and it's linked like the coil of the phone, right? And it exposed to the both extracellular and intracellular. And this is the most leading family of the validated drug targets in biomedicine. So as I mentioned, this is only small number of the type of GPCR. You see, this different type is HT, R7, CCR9, and everything. So every each of this receptor has been studied to see whether it really can be a drug target. This is just some statistics to see that the majority of GPCR is used to develop some of the drugs like opioid, dopamine, histamine, and others. Right? Yeah, any questions? If you have any question, just unmute yourself, okay? You can stop me anytime. Any questions before I continue? All good? Okay, then let's continue. The next type of protein is a structural protein. As the name suggests, is structural. So it's a protein that forms a skeleton, or it contributes to the mechanical properties of a living cell. So in a human, it's like the bone of the of us, because it gives us the structural properties, right? One of the example of the structural proteins called tubulin. Tubulin is crucial for the cell division and the cell mobility. And it's also crucial in the cell division because it works as a framework during the cell division, cell dividing process. If you see this beautiful picture, this if you can see my pointer, yeah, this one that I circle is one single cell. One cell, and inside is the blue color. What do you think is the blue color here? What organelle of the cell here?
Nucleus.
Nucleus, good. Is the nucleus. And what do you think is this beautiful green color?
The structural proteins, the tubulin.
Yes, the tubulin, because we're talking about tubulin. So it's tubulin. So it gives you the structure of the cells, actually. When you think about tubulin, it's only one protein, but it occurs like in the whole part of your cells, because it gives you the structure. It gives your cell the structure, right? It's a very complicated system, actually. And if you look at closely, if we see the structure of the tubulin is actually like this. One single subunit of tubulin, but it can form a polymer of tubulin. So this whole polymer, this is what you see here, the real image of the tubulin in the cells. So it's like a very long chain of a protein. Yeah. One of the application is in developing the drug for anti-cancer, the anti-tumor drugs. Is target the tubulin and inhibit the process of the cell division. So that drug, one of the most common drug, not common, one of the most known drugs called paclitaxel or Taxol. Taxol is an anti-cancer agent, we call it. It's actually a semi-synthetic analog of docetaxel. It targets the tubulin, because we're talking about tubulin, it targets the tubulin. What is the mechanism of action of this Taxol? It inhibits the tubulin depolymerization. So I mentioned before, actually the tubulin, it has some subunit, one single tubulin, and everything. But this one is called microtubule. This microtubule can assemble together, and it can form this long chain of tubulin, the polymerization of the tubulin. So what the Taxol do? The Taxol can bind to this tubulin, and it will inhibit the tubulin for the depolymerization, because the original of tubulin is the microtubule, right? It's like the single protein. It polymerizes into this long chain, and then after it's done, it can go back to these microtubules. The Taxol inhibit the process of this tubulin to become depolymerized. What happened then? When the taxol bind to here, so it makes the cell cannot reuse the microtubules, right? So it stay just like this. It stay in the polymerized structure. What happened to the cell? The cells later can go apoptosis and hence can lead to the cell death. So that's how the taxol acts on the tubulin, and these properties later can be used as an anti-cancer drugs to treat to kill the cancer drugs. Right, so we have talked about proteins, and the second biomolecules that we want to discuss here is the nucleic acid. So nucleic acid is also a functional biomolecules, we would say, but instead of amino acid, for protein case it's amino acid, nucleic acid is made of nucleotides, yeah? What are nucleotides? Nucleotides is actually just a unit. It has subunit, the base, whether it's pyrimidine or purine. It has the sugar, usually is the ribose sugar, and it has this phosphate, right? So it consists of these three units, again, base, sugar, and this phosphate. And these nucleotides can bind one to each other and then hence can make this a very nicely structured helix DNA. That's nucleic acids. And many important drugs also try to target the nucleic acid. For example, the antibacterial drugs. And the main classes of the DNA binding molecules or DNA binding drugs, they work on these four mechanisms. First is as a groove binders, intercalators, alkylators, and the DNA cleavage agents. We can check one by one, but for the interest of time, I will just discuss one of them. Let's see one of the examples here. One of the drugs that use to target those nucleic acid is called cisplatin. Cisplatin also is very common drugs you can find easily on the market. Cisplatin is actually one of the chemotherapeutic agent. It's not really administered to oral, so it will be administered through the IV injection. It's actually a small, very, very small and very simple molecules. It's a coordination complex of platinum, if you see here, and it targets the DNA. What is the mechanism of action of this drug to the target? It acts as an alkylating agent, actually. What does it mean? So it acts to the, when you have this DNA, the platinum here, the PT, will bind to the reactive center of the base called the purine, either the G or the A, right? Because it binds here, this covalent binding later can lead to the intrastrand and interstrand crosslinks and causing the strands to be broken. So let's see here. This is, let's say this is your DNA, and then when you are administered with the cisplatin, this side of the cisplatin can bind to your either G or E, and then it can make your DNA broken. Because the DNA broken, what happened to the cells? What happened to the cells?
Eventual cell death.
Eventually, the cell will be died. Yeah, correct. They will die. They will die. So that's one of the example of the drug targeting the nucleic acids. Okay, before we move, any questions so far from any one of you?
Doctor,
Yes, sure.
there's a question about this one of the example, which is the Taxol one.
Taxol.
Yeah.
Let's see. Yep, Taxol.
Yes, because this Taxol is like for anti-cancer agent, right? Because like right now the researchers are still researching about how to like need to reduce like the cancer. Is it like this agent is like just for increase the survivability of the patient or what?
Okay, okay. Even though now we have quite many of anti-cancer agents, right? But people still trying to look for a better agent or better drug. Why? Because apparently most of the cancer, they can kill cancer cells, right? But they not only kill the cancer cell, they also kill the normal cells. That's the problem. So the problem is that the selectivity of the drugs. We hope that the drug will kill only cancer cell. It will bind to only the tubulin, the protein on the cancer cells, not the normal cells. So that's the thing. So it doesn't really to what do you say just now, to increase the expectancy of the patient, right? We want to kill the cancer cells, but there always be a side effect because it's not only the cancer cells that are died, but also the normal cells. So that's why when you have this chemotherapy, right? When you are having these anti-cancer drugs during the therapy, sometimes you have the side effects because your normal cells also are killed, some portion of it.
Okay, understood, doctor. Thank you, doctor.
Thank you, Hezron. Any other questions?
Doctor?
Hey, Adam.
Yeah. I just want to ask about the monoclonal antibodies, if that's okay. I assume they bind to the cancer cells, I assume the glycoproteins on the top side, on the actual bilayer. But what happens afterwards? Do they just inhibit it, or do they cause apoptosis and cell death? How exactly does that process work?
Yeah, good question. And now the trend is shifted, as I mentioned, right? People used to develop only small molecules, but for the cancer, they move to these antibody-based drugs. So that's why you see just now there are many MAP drugs, right? This drug, we call it drug, but it's actually a very large biomolecule. If you compare the small molecule, like say this one, Prozac, it's a very simple molecules, right? You have a this, but when you talk about antibody is actually a part of protein, right? Protein is like, let me just go back. And where is it? Protein is like a very complicated, it's a very big. So when you compare the size of the small molecule drugs and the antibody drug, the size is like too much difference. What is the implication then? A few days ago, we learned that we want the drug to be able to be absorbed by the body system, to able to pass through the cell membrane. That's the problem with the antibody. Even though it has some specificity to some receptor on the cell target, but it has the problem with the cell permeability. So that's why when they develop this antibody-based drug, usually they will target the receptor on the cell surface. Here. Some of the proteins are inside the cells, right? The intracellular space. But the antibody-based drug, mostly they cannot pass through this system. So they will target the proteins on the surface of the cells. So what happened, usually, the antibody here, let's say this is antibody, yeah? Antibody will bind, interact with the system, with the receptor, and it will affect the functionality of this receptor. Similar things with these small molecule drugs, actually. Yeah. Does that answer your question?
Yes. Yeah. So when it binds, it would secrete something into the cell, or would it just inhibit the extracellular proteins?
No, actually, when they bind, let's say, because we have different type of receptor, it has different type of functionalities, right? When it binds here, it will affect the functionality of this very particular receptor. For example, it binds to the ion channel, for example, ion channel. The ion channel, the functionality is to pump in some of the ions that is important for your body, like sodium, potassium, from the extracellular to intracellular. But with the binding of this antibody to this ion channel, so the pumping of the ions in your body will not happen, right? It disrupts the functionality of this system. what happened to your cells? Your cells will have a lack of ions that is actually important for the biological process. So it works kind of like that. So the antibody doesn't really secrete something, but it binds to something that can disrupt the native functionality of that target. That's how it works.
All right. Thank you, Doctor.
Yeah. Thank you. Any other questions?
Yes, I have a
Before,
question.
yeah. Who's speaking? Cannot see.
It's Raihan.
Oh, hi, Raihan. Yes. Oh, finally, I can see your face.
Well, I was reading about the nucleic acid as drug targets on the book you showed just earlier, and I noticed 3 different methods. And what is the advantage of either one of them if the purpose is just to prevent the DNA from replicating?
Yeah. Are you talking about these methods?
Yeah, what is the advantage of either one of them if the result is we just want to prevent the DNA from replicating?
Okay, good questions. So the main goal is the same. We want to prevent the DNA from replications, right? And eventually it will prevent the cells for dividing, or it will lead to the cell death. Why we need this type of methods? Actually, these methods, because we don't really create these methods, but it based on the observation. So when we design some drugs, we see how is the mechanism. And apparently from the database, from different type of drugs, it acts differently on the DNA. Some of the drugs, they will bind to here, this groove binding, this specific area of the DNA. Some of them, they don't bind here, but they intercalate. Intercalate is just like they don't really make covalent binding to the DNA, but they just intercalate. They will be inserted in between. Intercalate, that's intercalate. And other things, they may act as an alkylator. They make covalent binding and they can alkylate the DNA. So based on that observation, the scientists then conclude, oh, these drugs, the main effect is to prevent the DNA from replicating, but the mechanism of action is different, depends on the nature of the drugs. So that's why.
Okay, thank you, doctor.
Thank you, Raihan. Any other questions?
Doctor,
Yes, Hezron.
yes, because for the latest slide, I mean the last slide
The last.
about the
Yeah. Is it this
chemical
one?
this cisplatin, yes, because the target is DNA. Why it doesn't want to target the RNA since RNA is like the copy of DNA?
Very nice question. Why you think so?
Because like from my previous during my high school, mostly the antibody science that will target the RNA since RNA is the easiest one because the DNA is complicated to be like target or to be like bind to it.
One of the reason is because they want to take the advantages of the structure of DNA. The main difference on the structure of DNA and RNA is, what do you think? DNA is double helix, right? And RNA is single helix. So the stability of this molecule is already different. So that's why it will target the DNA more. It prefers the DNA more because of the structure of this double helix of the DNA. That's one. The second one, we also need to talk about the abundance of this biomolecule in the cell system. DNA, we can say it's always there. The DNA is always sitting in your nucleus, like the king, right? So if you look at this cell structure, where is my cells? Here, inside your nucleus, but the RNA. The RNA actually is a product of transcription from the DNA. right? And it's not always be there. And when after it's done, the RNA will be synthesized based on the gene, not the whole RNA, only the gene, and then that's it. So that may be one of the two main possible reasons why it targets the DNA but not RNA.
Okay, thank you, Doctor.
Thank you, Hezron. Any other questions before we move to the next topic? All good?
Yes, Doctor.
Okay, let's continue for a little bit more. So we're talking about the target. We're talking about this drug will bind to the target, but what are actually the interactions between them, right? So now we know that the most drugs actually they interact through the weaker form of interaction known as the intermolecular bonds. So not everything, not all the drugs can make covalent bonds. Mostly they will have this weak interaction called the intermolecular bonds. What are they? They might be electrostatic or ionic bonds. They might be hydrogen bonds or van der Waals interaction, dipole-dipole interaction, or hydrophobic interaction. So the accumulation of this weak interaction, even though when you take individually, they are very weak. But when you take the sum of it, it will have some strength of the interaction that can allow the drugs to bind nicely to your target. So the interesting is that an equilibrium will take place between the drug being bound and unbound to its target, different than the covalent. When you have this covalent binding, like the C atom skeleton, right? It will just stay there. You cannot break it until you need to have some very strong forces to break the bonds, right? So you cannot do anything else. But for the drugs, because it's a weak interaction, it's intermolecular forces, it will have some state of bound and unbound. So these binding forces are strong enough to hold the drug for a certain period of time to let it have an effect on the target to produce this biological response. But it's also weak enough to allow the drugs to depart once the job has been done. It's one way of the mechanism of the drugs. So the functional group present in the drug we have discussed before, we call it the binding groups. Because when we see the structure of the drugs, you have these functional groups and other functional groups. Not everything have the binding affinity to the target, but those that has this binding affinity, we call it the binding groups. And the specific regions on the target where this binding takes place, we call it the binding region. So if you look at this cartoon here, let's say this is your target, right? The one in gray color. It has some specific binding site. And the drugs, this whole drug, they not bind, like all the functional groups may not bind to all part of the binding site. Only specific functional groups can bind. So these binding groups that actually is very critical for the efficacy of the drugs when you design the drug. So for this unbound and bound equilibriums, usually the drugs can bind to this target and it can produce some effect on it, and then it may be unbound again. So let's talk about some of the interactions, what do you call, the intermolecular interaction that can happen between the drug and the target. First is the electrostatic. For this, actually, I'm sure all of you are already familiar with this type of forces, but let's talk for a little bit. So electrostatic is the strongest type of the intermolecular bonds. We would say it is actually an interaction between groups of the opposite charge. So that's the requirement. It should have the opposite charge, such as the carboxylate ions and the ammonium ions. One is negative, another one is positive. So that's why it can have this electrostatic or ionic kind of interactions. In the drug-target interaction, if an ionic interaction is possible within this negatively charged and the positively charged functional group, it is likely to be the most important initial reaction or interaction when the drug enters to the binding site. This is one of the examples. Let's say the gray one is the target, right? Your target. The target, if it's protein, it has some functional groups from the amino acid. For example, it has these ammonium functional groups from the, let's say, arginine of the target. And then your drug has these carboxylate ions because it's the oxygen has this negatively charged and your ammonium, the ammonium site from the target has positive charge, so it can have this interaction. And this interaction, we call it the electrostatic interaction. Right? That's one. Another one, one of the common interaction is called the hydrogen bond. You are familiar with this already. It's also considered quite a weak interaction, electrostatic interactions. The hydrogen bonds, it happens between the electron-rich heteroatoms, the one that have a lone pair of electron, usually the oxygen or the nitrogen, and another one with the electron-deficient hydrogen. So we have this term for these two atoms, the one electron-rich and the one with electron-deficient hydrogen. We call it hydrogen donor. We learned a few days ago, HBD is a group that provides the hydrogen for the hydrogen bonding to be happen. And also hydrogen bond acceptor is a group that interacts with the hydrogen, right? But funny thing, interesting thing, I would say, there is a term we call the hydrogen bond flip-flop. It means that the functional group that can act as a HBD as a donor and also as an acceptor. So this is one of the example. When you see here the target, right? You can see my pointer here. The target has these atoms with the lone pair of electron. Because it has the lone pair of electron, it is more electron negative, right? So you have this electron negative, and then your drug may have these atoms with more electron deficient hydrogen. Because of this electropositive charge, it may interact with your target. And this kind of interaction, we call it the hydrogen bonds. Right. Just some of the notes here. The strength of the hydrogen bonds will depend on how strong the HBA and the HDB, right? A good HBA as an acceptor, it has to be electron negative and it has to have a lone pair of electron. And the common atoms that usually can have the hydrogen bonding acceptor properties is the nitrogen and oxygen, like this. So the oxygen have these two pairs of electron, lone electron. So that's why the oxygen can have two bonding. As compared to the nitrogen, it has one pair of the lone pairs of electron, and it can form one hydrogen bonding with a partner. Yeah, this is, I think I can skip this one. I think you can read it by yourself later. I want to talk about the other type of interaction called the van der Waals. It's also considered a very weak interaction. It usually happens between the non-polar region of the molecules and are caused usually by the transient dipole-dipole interactions. So this involves the interaction usually between the hydrophobic regions of a different type of molecules. So such as the aliphatic substituents. Aliphatic means the C carbon, yeah, or overall the carbon silicon. And for this to happen, for the van der Waals interactions to happen, the drug has to be close enough to the target binding site because it's very weak interaction, right? So the distance between the drug and the target should be close enough so that the interaction can become considered to be important. This is one of the example. It happens because if you move it a little further, the van der Waals interaction may not happen because the dipole-dipole will not be induced. But when it's close enough to the binding site, so the dipole-dipole interaction might be induced by this whole hydrophobic skeleton, and that's why the van der Waals can happen. I mentioned about dipole-dipole. I think you already know about dipole-dipole interaction as well. So the molecules can have a permanent dipole moment resulting from the interaction, from the electronegatives of the atoms and the functional groups present. For example, the ketone functional groups, it can has the dipole moment due to the difference of the electronegativities of the carbon and the oxygen that making up the carbonyl group. So when we're talking about this drug and target interaction, this binding site, the binding site from the target may also contain some functional groups, right? And it will have a various degree of local dipole moments. So the dipole moments of the drugs and the binding site later can interact when the drug approaches, right? So this intermolecular attraction can happen. This is just some of the example, but I think I can skip this. This is like the dipole because, okay, let's see one example here. You see this has your drug has this carbonyl group here because it's carbonyl, the C atoms will have more electropositive charge and the oxygen will have more negative charge. This electronegative charge will interact with this ammonium ions from the binding site, and it will make this interaction that we call it dipole-dipole interaction. I think it's the last one, hydrophobic interaction. I think it's easier to remember because it's hydrophobic interaction. Simply, it means the interaction between the hydrophobic regions, right? These hydrophobic forces are important when the nonpolar regions of the molecules can interact. So we only talk about this charge, electronegative, electropositive interaction, but actually the neutral species, the neutral molecules, they may have also the interactions, what we call it these hydrophobic interactions. Okay, next one maybe is considered not a very important interaction, not really a popular intermolecular forces, but it happens actually. It can be considered, it should be considered in the drug and target interaction. It's called the water interactions. Why is that so? Because we know when we design a drug, we target the drugs to produce the biological response in our body. And our body actually exists in a aqueous environment, right? So the drug has to travel through this aqueous environment in order to reach the target in the body. So both the drugs and the targets, they need to be solvated with the water molecule first before they can meet each other, right? And the water molecules surrounding the drug and the target binding site will have to be stripped away before the interaction can take place. So it's one of the weak interaction that can help to bring the drugs to the binding site, not so much on the drugs and the target interactions. Right, any questions so far?
I have a question.
Yes, go ahead, Raihan.
Considering all of these interactions, that means if we want to make an effective drug that binds the target, then we clearly need a thorough knowledge of the binding site, correct?
Correct, exactly.
Then how do we know, then how do we properly map the binding site of our target location?
Very nice. So it leads me to the next slide, actually. So first, we need to know the target first, right? And the target can be protein, let's say. And in the protein, let's say this is one structure of protein here, yeah, in the blue color. One protein. We don't know yet if it's very new protein, is a newly discovered protein. We don't know which one is the binding site, which one is the catalytic site. We don't know yet, but we can do it by trial and errors usually. So if the target protein that we discover is not very new, it's already been studied before, so we can have certain degree of understanding on where the binding site would be. For example, let's take this as example. So this is from my very, very old paper, 2010. So here we try to design a drug as an inhibitor for dengue, So this protein is actually the protein from the virus, and this is the drugs that we design. So when we do this computational simulation, we call it the molecular docking, we can know that, oh, the drugs bind to this area, right? That's from one drug. And then we can do the same thing. We can do the molecular docking to say thousands of compounds. We can do the binding, the simulation, and then we can see where the drugs are preferred to stay. So from this case, actually from hundreds of compounds, the drugs almost always prefer to have binding here. So we can say, oh, maybe this site is the binding site because the drug prefer to be bound to that site. From this information later, then we can learn what are amino acids that contains in this area. So for example, here I can take some different look at this. If we remove the other part of the protein, right, because it's not really our interest anymore. We want to see only in this area, the binding site. Then we can know, oh, the protein, not the protein, no, the amino acids that interacts or may interact with the drugs is this one. For example, aspartate here, tyrosine, serine, and histidine. So from this kind of experiment, we can estimate where is the binding site. Does that answer your question?
Yes, thank you, doctor.
You're welcome. So this is one of the example on how we can do a rational designing of a drug. So we can, design the drug molecule first. We can design hundreds or thousands, or whatever the numbers you like. And then when we cannot yet do the experiment in the wet lab, because it takes a lot of resources, we can do the simulation first on the computer. So we have this target, and then we can try to do the binding, the digital binding, I would say, from all the target, from all the drug molecules to the targets. And then we can do the analysis on how the drug behave or preferred on the binding site. For example, this one, I have this the best candidate because it has the mathematically, statistically, it says it has higher binding affinity to this site. Then we can see how the drug prefer to bind to the cells, to the target. So it's very interesting if you see. This is my candidate, right? Now from this kind of schematic representative, I can say, oh, it can be the best candidate. It prefers to be binding here because it has some functional groups that can match or can fit in this pocket of the protein. The same thing as this one, the same thing as this one. And then from there, you can learn on how is the interaction happen. For example, here I have the red color here is actually the oxygen, yeah, if I'm not wrong. It's the oxygen. So you can learn it has the oxygen, it is electronegative, and you can find out what kind of amino acid are here. Then you can predict the interaction happen there. By using this information, later you can optimize your drug molecules. For example, from this side, you say, oh, okay, I know this amino acid here is more red, it means more negative, let's say. It's more negative. So you need to make the compound here to be more positive. Then you can optimize your protein structure, your drug structure, your drug compounds, so that it can fit better to this binding site. So you can have a best possible candidate for your drugs. That's how it works. Any questions again? Anybody wants to offer questions or opinion, or you can ask anything? No? No questions? All good?
All good,
Okay.
doctor.
All good, doctor.
All good. Okay. Then so we've come to the very last slide from this lecture. So it's one of the example of what I call the molecular docking. So in this case, it's the same thing with the previous slide here. So here we try to design this drug, and then we do the docking to the target. And then from the analysis, we can find the binding site and the binding interaction between the drugs and this binding site of the protein. So if you see here, wait, let me find my pointer. Yes. So this is the drug molecules. And the dotted here is actually the binding site, right? The binding site of the target. And in the binding site, you have the information of the amino acid nearby the binding site, like this ASP, aspartate, cysteine, serine, lysine, and everything, right? Why this compound sits here? It's because they must have some interactions, right? You already know. For example, here, for example, this one, you see my pointer, right? I point this oxygen, right? We already know this is oxygen, this is a double bond, so it's carbonyl. In carbonyl, the oxygen here must be electropositive or electronegative. What do you think? What is the possible charge of this oxygen here? Oxygen, we know it has 2 lone pairs of electron, right? So it must be more on the electronegative. It's more negatively charged. It sits here and it actually interacts with these amino acids called arginine. You need to check what is the structure of the arginine. Now we know the arginine has this NH2, right? The amines. The amines we know is actually slightly positive. So you know why these drugs preferred here, because they make interactions, and one of the interaction is this oxygen make interaction with this arginine through, for example, maybe it's hydrogen bondings, or maybe it's electrostatic interactions, right? It can be both. So another assignment to you, because I have to give you assignment here, can you identify all the possible interactions happening between these functional groups of the candidate of drugs with the neighboring amino acids? Okay? Is it clear enough?
Clear, doctor.
Good. Okay, so that will be
Doctor?
your... Yes, who's speaking? Hazard?
Yeah, Hezron.
Yeah.
The blue spot is also the binding site, is it?
The blue spot here?
Yeah. There's a few of blue spots also. The binding site, is it?
The binding site is actually the dotted lines.
Okay.
And this blue is actually just to tell you that it may contribute to the interaction between the drugs and the binding sites.
Okay.
This is actually
Understood.
the real analysis, the result from the docking experiments. Yeah. So you can consider all types of interaction. Yeah. Not only the hydrogen, the dipole, the ionic, but maybe hydrophobic, right? The hydrophobic. So you can tell, for example, here is the benzene. Benzene is hydrophobic, right? You can check the neighboring amino acid, whether they have hydrophobic so it may interact, something like that. This one is easy, the one in green arrow, because I give you some. It's cheating, but it's okay. You know, it's OH, right? Hydroxyl, and this is the serine. So you know this is maybe hydrogen bonding happening there, right? It's very easy, but you can identify the other type of interactions here. All right, you may take a picture of it. You can screenshot this one so you can do your assignment. I give you some time to screenshot, everyone, please. Okay. All of you has taken the picture here, have been screenshotted this slide. Can you give me your
Yes, Dr.
thumbs up?
Already.
Oh, nice. I see a girl now. Husna, thanks. Can you give your thumbs up if you have screenshotted this slide? So I know that every one of you already done it. Good.
All good.
Okay. Okay, not everyone, but okay, I see majority of you.
Doctor,
Yes, Hezron.
I do have a question
Sure.
because for binding site, is it I mean for all the amino acids here, right? Is it like is there any effect on the pH and the polarization itself? Because some of the amino acids is a non-polar and a polar amino acids. So is there
Correct.
any effect and with the pH also, with the binding sites?
Yep. Because we consider this one, this is happen inside our system, right? In body system. And the pH should be our physiological pH. So when you consider this drug and target interaction, we always consider this at the physiological pH, which is 7.4, about 7.
Okay. So how about the polarization? I mean for the polar and non-polar. Is like maybe there's a effects on that?
Because we have this specific pH, I think it will not make much difference. Yeah, because you consider this, the pH is about 7. So you can check from the literature, like what is the charge of this amino acid in the pH of 7. You can check that.
Okay, understood, Doctor. Thank you, Doctor.
Thank you. Anybody else also have questions or comments or anything? I think this will be our last lecture with me, not our last lecture from this series, because you will have another, I don't know, 7 or 8 lectures, right? Okay, if it's all good, then
Doctor.
We've come to the end of our lectures. Who's speaking? Adam.
Me, yeah.
Yes, Adam.
I'm just wondering, is there a minimum amount of like page that can be used for this? Because in the assignments above, because there's of course a maximum of 1 page, but of course depending on if we get to the point very quickly, then we just have to
I think for this
waffle
one,
on after that.
yeah, the first assignment was 1 page, right? This one also maximum
Yeah,
1
maximum
page.
of 1 page. Yeah.
Just 1 page, yeah. Yeah.
Okay, just 1 page then. Just a full 1 page.
Yeah, 1 page. It's easier. Yeah, it's easier for you and me.
Okay. All right. Thank you.
Oh yeah, for this assignment, could you all please, you also need to include this figure in your page, right? So you have this figure, just make it smaller in the top of your page, and then the bottom you can just write down the possible interactions that it's happening. So it's easier. You can point to that picture. It's easier also for me to check your work. Okay. If no more questions, then maybe I will give it back to Dr. Dewanga. Thank you, everyone.
Okay, Dr. Samira, thank you very much. Please give physical applause if you can so, or give thumb up for Dr. Samira. Okay, thank you for very interesting and interactive lectures. It's really comprehensive study, not only talk about chemistry itself, but it combines research field among chemistry, medicinal, and as well as biology. Okay, once again, I still have one chance for students who have questions or comments, whatever you think now and whatever you will comment. So please ask and please show yourself. Just make it like your class in University of Malaya, because we only record only just 3 or 2 students who are active in the participation of this lecture online. How about the 13 students specifically for Malaysia students? Please, if there are any questions, please show yourself. Is there? Once more again. No, you only have this chance with Dr. Samira, because as Dr. Samira says, we will not have a lecture online with Dr. Samira again. Okay, no? Okay, it's okay. Kindly note that today, Adam, you are set. This is the last opportunity to have a lecture with Dr. Samira. It's no problem. You will come here and you can meet Dr. Samira directly. Okay, please remember, I'd like to remind you, it's okay to you to not open your camera today. And for next lectures, lecture 4 next week, please open your camera so we can assess completely your attendance, because other filling out the attendance form, you should open your camera to show you are active in the lecture session. Okay. And second, please be more active in the discussion during the online lecture, because we will consider and we will make some recognition and also award for the best participant. So please consider it for the next lecture. Okay, if there is no question as no discussion again, thank you all for your active participation in today's lecture. I hope this session has provided you with a better understanding, of course, for the basic concept and also the implication in drug discovery and also development. And for importantly, it can be an additional understanding for your research at future in the University of Malaya. Okay, once again, I would like to extend my sincere gratitude to Dr. Samira for your enlightening presentation. Your expertise and passion for this subject were evident throughout the lecture, and the insight have undoubtedly, of course, enriched our understanding of drug target and their interactions specifically. Okay, lastly, thank you again for your attention and for listening, and I wish you all continued success in your study, specifically for our next lectures. Okay, this is from me. I will give back this chance to Miss Hilna. Thank you very much, Miss Hilna. Bye-bye.
Thank you very much for our moderator, Bapak Dewangga Oki Bagus, SSI, MSI, PhD. And thank you very much to Ibu Samira Hussein, Alamundi, PhD, for your insightful and comprehensive lecture on drug target and drug target interaction. Your expertise and clear explanation have greatly enhanced for our standing in this field. Ladies and gentlemen, now we come to the end of today's agenda. But before we conclude, we will take a moment for photo session. The photo session will be guided by Rohman. For Rohman, time is yours.
Okay, please open your camera. Okay, 2, 3. Okay, again. 1, 2, 3. Okay, thank you.
Thank you very much for Rohmat and all of the participants in this photo session. Before we part way, I want to remind next lecture session about participle of pharmacological and ADME with Bapak Dr. Apoteker Catur Jarnika MSI and will moderated by Bapak Dr. Bayu Ardiansah SSI MSI. So please make sure to join us at the same time at Tuesday next week. Thank you very much for your kind attention. Have a great day and wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Hilna.
Waalaikumsalam
Thank you,
warahmatullahi
Dokter.
Thank you very
wabarakatuh.
Thank
much.
you, everyone.
Terima kasih,
Thank you.
Dr.
Good day ahead.
Samira.
Thank you,
Thank
Bapak
you all
everyone.
participants from UI and UM especially.
Yeah. I hope I can see you all when you come here to Indonesia, yeah? Those from UM. Yeah.
We will, Doctor.
Okay, see you
Most definitely.
everyone. Have a good
you.
day.
Thank you, Doctor.
Bye.